All right, hello, and thank you for joining us for Chardan's Virtual Ophthalmology Summit. The focus of this current installment is on rare ophthalmic diseases. My name is Daniil Gataulin. I'm one of the senior biotech analysts here at Chardan. It is my pleasure to introduce Stephane Boissel, CEO of SparingVision, and Kelly Stasi, Chief Medical Officer of SparingVision. Stephane, Stasi, nice to meet you. Thank you for joining.
Thanks for having us, Daniil.
Of course, our pleasure. So the format for this session is going to be a fireside chat. So with that, let's go ahead and get started. To kick things off, Stephane and Colin, maybe you can spend a few minutes to introduce yourself and provide a brief overview of SparingVision and key programs.
Yeah, maybe Kelly, why don't you start with an introduction of yourself, then I will introduce myself and then the company.
Great. No, thank you. And very nice to be here and talk about my recent. I recently joined SparingVision. I'm very excited to be part of the team working on such an amazing scientific program. And with great people. My background, I'm in ophthalmology for about 30 years now. I'm an ophthalmologist, a physician scientist, priorly in academia. I was faculty at UPenn before I joined industry, initially at Novartis and then at smaller biotechs working on clinical development for gene therapy and cell therapy programs. And I'm very excited to join the team since about a month ago. So I'll get it back to Stephane.
Thank you, Kelly. So I'm Stephane Boissel. I'm the CEO of the company. I joined in 2020 from Sangamo Therapeutics, where I was head of business and strategy in San Francisco. I've been in this industry for 25 years in various C-level on board capacity. When it comes to SparingVision, so we are a spin-off from the Institut La Vision in Paris. We are a clinical stage genomic medicine company with a focus, obviously, on the ocular space. We've raised to date a Series A and a Series B, accumulating 130 million euro. Our lead investors are Jato from France, UPMCE from the US, BPI, 4Bio, Ezios, and the RD Fund from the FFB in the US. We are really focusing on developing breakthrough treatment for retinal disease with an initial focus on retinitis pigmentosa, which is the leading form of IRDs, as well as on dry AMD. Our most advanced product in the portfolio are gene therapy product, and I guess this is where we should focus today. And in gene therapy, we like to say that we do it in a very different or distinct way from our peers. First, all our products are first-in-class therapeutics with no competition on the MOA. They are all, and this is a very important feature, agnostic to the causative mutation or to the origin of the disease. And they are also independent of the disease. If the biology works, the product will be working in many different ocular diseases, as long as, of course, we find disease with the similar or the same physiopathology. In addition, to complete the introduction on our gene therapy product, we've made the strategic decision to focus on prevalent indication, and for which there is no effective standard of care. And we believe that the combination of the strategic decision we've made, as well as the technical feature of the product, will help us to solve the return on investment equation that many players in our field are currently trying to and struggling to solve, actually.
Okay, excellent. Thank you, Stephane. Thank you, Kelly. Before we jump in and talk about your program and your products in more detail, let's talk about the lead indication first. So you mentioned retinitis pigmentosa. Can you tell us about the disease, how the disease presents itself, various stages of RP, And specifically, what stages of the disease do you expect to be able to target with your product?
Daniil, retinitis pigmentosa, as I said, is the leading form of inherited retinal disease. You are talking about something like 70% of all cases. It's a population that has an impact of 1 person out of 4,000. It's approximately 100,000 people in the US and more or less the same in Europe. And it's a disease which is highly heterogeneous and which is caused by something like over 80 different genes with like 250 different mutations. And it progresses in stage, and it begins generally with the degeneration of rod photoreceptors, which is leading to night blindness and to loss of what is called the peripheral vision. And then over time, the cone photoreceptors, which is the other subset of photoreceptors, are started to be affected. And since they are responsible for central and high acuity vision, this part of the vision start to degenerate as well, eventually leading to blindness. And this progression, in most cases, will occur over decades, which is offering a very broad therapeutic window for intervention. And as I said earlier, there is currently no standard of care for this disease. There is one product which is approved, which you know well, which is called Luxturna, but which is approved for a very small subset of patients on a very early onset indication with a very specific mutation. So in this disease, again, we come with a very different approach with 2 different products, soon a 3rd product to be developed. A first product that is going to go and work at preserving the cones from dying. So it's really about function or vision preservation. And then the second one, which is about restoring function. And for the first one, to your question, the idea for us is to intervene from early diagnostic until almost the end stage of the disease. The key for that biology to work is to find cones to be preserved. So of course, the sooner you intervene, the more you will have to preserve. But that's really something about this first product in particular called SPVN-06 is that the window of intervention is very broad.
Got it. Okay. And in terms of the approach itself for RP specifically, why do you believe it's particularly important to develop a gene agnostic approach?
Well, it's very simple, and it's very unfortunate for the patient affected with some very small mutation. But if you want to develop a gene therapy or gene therapy specific to all indication, you will have to develop something like 250 different products. And nobody will ever do that. And especially now that it's more and more challenging to find money for gene therapy, and even more so for gene therapy addressed in rare disease, this is not possible. So we have to find a different way to do it. And this is where we believe being gene agnostic or mutation independent becomes very relevant because, again, our product, the biology, is expected to work independently of what has caused the disease to start with. So for the cost of development of one product, we will potentially treat all comers and have a very significant commercial potential. So this is, again, solving an equation which is very difficult to find or to solve today, which is the ROI equation in gene therapy. And so we believe, again, being gene agnostic is the only way to solve this equation and to address the huge unmet need.
Got it. Yeah. Okay, that makes sense. And so there are several approaches that are currently in development for RP. Some even include optogenetic therapies for later stages of the disease where patients lost all or majority of their vision. With your approach being so novel, with no competition and no precedent in the clinic. What gives you confidence in the program's success, either from preclinical data or from the mechanism of the product?
So there are many questions in your question. So first, we have to make a distinction between the first two, SPVN-06, which is currently in phase 2 in RP, is not about function restoration, and the competition, so to speak, is not optogenetics. SPVN-20, our second product, which is about to start phase 1, is indeed comparable to optogenetics in the sense that the biology aims at restoring function, okay? But we believe that in this space, it comes with a significant competitive edge in a sense that it will target the cones, while most optogenetic solutions out there, they will target other retinal cells, such as ganglion cells or bipolar cells, which are cells which very function, given by nature, is not to do what the cones are supposed to do. So by restoring aiming at restoring function in cones that have lost their ability to function, we believe we are targeting the right, cell population. And importantly, the cones, as you know, are naturally sensitive to light. So we don't need, or we believe that we won't need to use, a light stimulation device in association with the product. So it's a very elegant biology, very different from the other optogenetic solution, and we believe it has a very significant competitive edge. Now to the second part of your question, the reason to believe. Obviously, if we've been successful so far in raising a lot of money, and if we've been successful to get to where we are with the two products, it's because we have some very good reason to believe it will work. And most of the reason is for the first product, SPVN-06, 20 years of academic research with a very vast corpus of preclinical data. Then 2 years ago, we started a phase 1. We have now established the safety profile for SPVN-06, and the safety profile, and Kelly can speak to it in more detail, is so far very good. We've started to observe some, let's say, early signals of activity, although it's too early to conclude, but this is kind of confirming that we are in the right direction. And importantly as well, recently, as you know, we started to develop this asset or this product in the clinic in RP, but we went to see the FDA. We went to see the FDA through a Type C meeting and asked the question of whether, with the non-clinical package that we have in RP and the safety profile that we've observed from the first in-man study in RP, can we transition directly and explore SPVN-06 in GA, geographic atrophy secondary to dry AMD. And apparently the package that we proposed to the FDA was convincing enough, and we were given the go to go straight into GA without having to redo a non-clinical package or without even having to redo a dose escalation study. So all that to answer to your question on the reason to believe, again, 20 years of academic research for the first asset, something like 10 for the second asset, and for SPVN-06, which is in the clinic, already a well-established good safety profile, some early signs of activities, and the confidence from the investors, the investigators, and the regulators that what we are doing, makes sense.
Okay, yeah, it makes sense. To get this out of the way, and let's spend a couple of minutes on technical aspects of SPVN-06.
Yeah.
So this construct carries RDCVF and RDCVFL transgenes. Can you talk about what these transgenes are and what specifically is their role in RP, and now that you've expanded the program, we're planning to expand the program into GA as well?
So maybe, Kelly, if you are back with us, can you speak about the biology of RDCVS first? Yeah.
Sorry, thank you. And apologies, my computer decided to restart on its own. So these are very well-known, naturally occurring neuroprotective factors that they are secreted by the healthy rod cells, and they support the viability of the cones. The RDCVF VF promotes the glucose uptake in the cones and overall supports the metabolism and the survival, of course. And the RDCVFL, it protects against oxidative stress because it acts as a thioredoxin-like antioxidant. So one of the beauties of this program is that it combines the two. So together they replicate the natural support that the roads provide to the cones. So in RP, as the roads degenerate, this support is lost. And we have seen actually that this is similar to the GA pathophysiology. And SPVN 06 can restore that support, helping the cones to survive longer and potentially delaying or even preventing the loss of the central vision, which is the most useful part of our vision for everyday activities like reading, for example.
Got it. Okay. So yeah, okay, that makes sense. So let's shift gears and talk about the clinical program that you have. So SPVN-06 is in phase 1/2 Prodigy trial in RP. You've presented some, you shared some safety data from the program. You've had some periodic updates there. Before we talk about those, can you remind us what the design of the trial looks like and what endpoints specifically in outside of safety you're looking at?
Yeah, absolutely. Oh, sorry.
No, Kelly, please go ahead.
Thank you. So Prodigy is a two-part trial, as you all know. It's an open label dose escalation first and then expansion. So the phase 1 part included 9 patients with severe late-stage retinitis pigmentosa. And it was focusing more on the safety part and the dose finding of the maximum tolerated dose. The second part of the study expands to a total of 24 patients which have less severe disease, intermediate stage of RP, and includes 2 active doses and an untreated control arm. Now, all these patients are going to be followed, of course, for 5 years after dosing, both for safety and efficacy. And we expect to start looking for real efficacy endpoints at least after 12 months of dosing in the second part of the study, where we have a more expanded population with less advanced disease. The key endpoints that we are looking at are very well defined in this general population. We are looking for best corrected visual acuity, low luminance visual acuity, retinal structure, and other functional endpoints like microperimetry, also like quality of life. We aim to see slower progression of the disease, like slower degeneration, and possibly preservation of the functional in the functional endpoints.
Got it. Okay. So go ahead.
No, go ahead. I'm done.
Okay. So for the part 1, the more advanced patients. So all these patients receive treatment in one eye, correct? Not the second eye has not been treated. So second eye would serve as a control, as an internal control. So for the more advanced patient population, and given the mechanism of action of this approach, you don't expect it to restore vision, but you expect it to slow degeneration, correct? With that caveat, what would the ideal efficacy readout look like to you from the more advanced population?
So it will be tricky to tell because this is an advanced population, as you said, and it will be also a matter of like how long it's gonna be. It's gonna take to see the results. It's only 9 patients, and with increasing doses, and we are gonna be looking for an overall signal, at least after 1 year. It's we are very happy to see so far that we have no issues with safety, because that was the major outcome of the that we were looking at from the first part of the study, and we are very happy to see that there is no issues with inflammation or immune responses or any serious adverse events, or no dose limiting toxicities, no trial discontinuations. And in the second part of the study now, with intermediate retinitis pigmentosa, we are very happy to see also that the preliminary safety data from the first 2 patients look very consistent with the first part. They have a very favorable safety profile. So that's very encouraging, and we are gonna be keeping an eye for more readouts outside of safety on efficacy as we move along.
And if I may chime in, so Daniil, we've always guided to the fact that we were not expecting to see much in terms of efficacy from part 1, given how advanced, unfortunately, the patient population was, and given how slow the progression of the disease is in this population. So to your point, and without being able to quantify today, most of what we expect in terms of efficacy readout will come from the ongoing part 2 that we are in the middle of recruitment of, and that we expect to be fully enrolled within the coming, let's say, weeks or months. And it's, again, I think it's too early to quantify what we are expecting. It's not, the study is not powered statistically to give us anything. So we will be really looking for anecdotal evidence. And you raised the point earlier of the control. Keep in mind that in the part 2, we have a control arm, which is, so it's not the fellow eye. It's an arm in which 6 patients will be randomized. They won't receive any placebo. There will be no sham injection. They will be just observed like in a natural history study. And in addition to that, we have an ongoing prospective natural history study with more than 80 patients. We are in the year number 5 of that study. And that study, plus what we will observe from the part 2 patient, will help us to quantify what we expect to see eventually as a benefit. But it's too early to tell.
Got it. Okay. Given the mechanism of action of SPVN-06, do you potentially expect to see any dose response in the part 2?
So maybe, Kelly, you want, well, first we have only 2 doses for the part 2, but please.
Yeah. It will be questionable, and I don't think we can actually answer this question about really seeing dose response yet. And if we expect one, it will be tricky to say even with this number of patients yet. But we'll be looking for one, but we don't know if we are gonna see something.
Okay. And another follow-up question to data interpretation. This goes back to the intro or the overview of RP. RP being a very heterogeneous disease, some patients progress faster, some slower. Are you looking at, or do you have an idea of certain genotypes being associated with different progression rates? And are you looking at different genotypes or paying particular attention in the phase 2 study so that you can make those distinctions?
I can take this one if you want, Kelly. So we have, in the natural history study that we've conducted, the prospective one, which is mimicking the Prodigy clinical study in terms of the genotype. We have 3 genotypes, RHO and PD6A, PD6B. And so far, we have not seen any difference in progression in markers of activity based on genotype. So we need to find elsewhere what will make some patient faster progressor than the other, because obviously that will be a population of interest. But again, so far we have not seen any difference based on genotype. Yeah. Anything else you would like to add, Kelly, on this?
Yeah, I would like to say that we will keep looking though, because in theory it's supposed to be, as you mentioned, some genotypes progressing faster than others. So this is one of the things that we have planned to look for during our analysis. So stay tuned as more data become available.
Got it. Okay, and so in terms of the next steps and the current status, so obviously the phase 2 part is enrolling now. Have you provided a public update on the enrollment status, and when do you anticipate sharing primary endpoint data from this program?
So I can speak to that. We are, as I said, in the middle of the enrollment in the part 2. We expect to be fully enrolled in the next, let's say, few months. And then for the data, it will be 12 months after the last patient has been dosed. So you have to assume that the data will be, because we need time to process, analyze, and so on and so forth, sometimes in Q4 of 2026.
Got it. Okay, awesome. And with respect to regulatory interactions and just the regulatory landscape, so we've seen recently in the past several months, I guess, in the IRD space in particular, some regulatory flexibility from the FDA and willingness by the agency to potentially lessen the burden, the regulatory burden in rare diseases, including some examples of allowing seamless phase 1 to 3 designs instead of asking for separate phase 3 programs. Do you have, I know it's a little early maybe right now, but do you have any idea of if you will be able to transition this into a seamless phase 1 to 3 study, or do you think you'll have to have a separate phase 3 program?
So actually, it's a very topical topic for us, subject for us, as we want to start registrational studies in 2027. So I will leave it to Kelly to answer, but this is definitely something for the 2 most advanced programs that we are seriously thinking about. Kelly, you want to speak in more details to the flexibility that the regulators are giving us?
Yeah, and we are actively considering different options right now to see how we can overall accelerate the program. But currently, the phase 2 is designed with some pivotal elements, but we may need a phase 3. We are looking into a really ambitious clinical development strategy. And we try to actively leverage all available regulatory pathways to accelerate the progress of the program and maximize the impact. So once safety is proven and preliminary efficacy signal is seen with both actually the programs that we have, our goal is to go straight into registrational study. But of course, that will be based on the regulatory input, and but we'll try to maximize the potential for accelerated approval. So we are trying to move as fast as possible.
Got it. Okay. So let's shift gears and talk about your other program, SPVN-20. So that's in development for later stage of RP. I think if you break it down, would be stage 3, 4. That product carries GERK transgene. Can you talk about this approach and the importance of GERK in being able to potentially restore some of the vision in RP population?
Yeah, Kelly, you want to speak to that?
Mm-hmm. Yeah, no, gladly. That's a program that I'm very excited about because this is designed to improve visual function in patients who have, as you mentioned, advanced RP and they are practically blind. So these patients, they have still dormant cones, which they lack the outer segments, but they retain the inner segment function. So we utilize a G protein coupled ion channel, GERK, to stimulate an alternative phototransduction cascade within the still alive inner segment of dormant cones and make them photosensitive again. So that's a fascinating biology, to me at least, and I think to many people, and it should be able to deliver results very rapidly after the transgene is expressed in cones. So we are getting ready. We are very excited to be getting ready to inject the first patient, to treat the first patient soon, and we hope that we will be able to see both safety and efficacy data in the next year.
Mm-hmm. Okay, that's excellent. So another distinction, technical distinction for SPVN-20 versus SPVN-06 is that 20 is, I believe, intravitreal, right? It's administered intravitreally. SPVN-06 is subretinal. What is there mechanistic rationale for going about it this way? Is there a difference in capsids? Is there a difference in more underlying biology that led you to this decision or the strategy?
Kelly,
Yeah,
you
the underlying
that?
biology, yeah, no, thank you. The underlying biology is critical here because this population is much more advanced and they have a fragile retina. So this is not a retina that you want to stretch with a subretinal injection and traumatize it further. So it was critical to have a less traumatic approach of such as an intravitreal injection. So I'm very excited that we have a way to actually help these patients in the best way possible. And we have, there are data on the intravitreal injection in non-human primates with very strong expression in the foveal cones after injection. So that's a population of cells that we want to target, and it is much. It is very obvious that they are targeted very well with the IVT injection. We are using, since you asked, a different capsid, and yeah, a different capsid than the SPVN-06. It's still a bioengineered capsid, and it looks like it's working very well for targeting the cells of interest. So we are very excited to get to humans and get the results that we are looking for both safety and efficacy.
Got it. Okay. And SPVN-20 being intravitreal, do you need to employ a special prophylaxis regimen to potentially reduce any instances of inflammation?
We do employ the standard tried already in other IVT injections prophylactic regimen of steroids. So we don't expect to have any surprises there, since there are there are already published results of very successful prophylactic steroid regimen.
Got it. Okay. And another question. So the mechanism of action of SPVN20 requires the presence of cones, right? They're dormant cones, but they still need to be there. How do you screen, or how do you determine that patients actually do have those dormant cones? And is there a threshold of how many or what fraction that they should have for this to potentially be effective?
Yeah, we evaluate the patients with OCT, of course, which we are happy to have nowadays very detailed OCT evaluation, and we can really see that the cells, the patients that we enroll, they still have cone cells viable. Prior studies, they have shown that about 60% of patients with low vision, they still have dormant cones that can be seen in OCT, as has been reported by groups both at the Institut de la Vision and at the IOB in Basel. So we do enroll patients who still have viable and dormant cones.
Got it. Okay, I understand. In terms of the next step, so the phase one Nirvana trial, right? That's about to start. You mentioned that you're about to begin dosing your first patients. I imagine this is going to be a standard 3x3 design first, with the expansion later. And for that first part, are you looking at patients with no light perception, or do they have any residual vision?
So currently the trial has no light perception patients, and we are definitely considering like expanding to patients who have some low vision remaining in the future.
Okay. And what would you consider to be a home run data from that early from those early patients?
I can take this one,
Yeah.
Kelly. So it will really depend at the end of the day, Daniil, of the population. If you consider a no light perception patient and you move this patient from no light perception to light perception, this is a home run. And then if you move a person who is only seeing hand motion and you move that person to counting fingers, that's a home run. So it will really depend on what the population will be, at the end. But as Kelly was saying earlier, the beauty of this biology is that we should know, very, very rapidly whether the biology is working after the transgenase express. In a matter of months, we should be able to observe, a difference. So it will be, the home run will be a rapid gain in something that the population has lost, and it will depend again on what the population has lost.
Okay, makes sense. So we have a few minutes left. I wanted to quickly touch base on SPVN30 program. That program combines the attributes of 06 and 20, right? It combines the transgenes from those programs. And can you tell us what the potential next steps are for this program? Are you waiting to see some further data or more advanced data from Prodigy and Nirvana before advancing SPVN30 into the clinic? When might we get an update of the next steps there?
So obviously we need to prove the biology of 20 and 06 independently before thinking about 30. But at this stage, nevertheless, we are looking at two things. We are looking at the technical feasibility, because as you said, we are going to combine or put a few transgenes into, possibly one single AAV or one single vector. And we need to find the right promoters. We need to, we have limitation as to the, cargo size. And then obviously we need to make sure that the two biology are synergistic. So not only that they work independently, but that together they will, provide the effect that we would like them to provide. So this product is clearly not ready for clinical showtime. We have some time to build a strong, technical momentum, and then to prove the concept in some animal models before we can invest massively. Clearly, the priority of the company, Daniil, for the next 2 years is to prove the concept in the clinic of 06 and 20, and then it will be about time to think about the rest.
Got it. Okay, that makes sense. Just a couple more general questions. As you're advancing those programs forward, how are you thinking about the CMC strategy, and is the product that's being used in those early trials, is that going to be the same product that you will use in the later trials and then potentially commercially?
Yeah, so we have a very strong CMC group within the company. So we've made sure that very early on we were investing we were doing what we had to do to make sure that we were not going to lose time, eventually. So first, in terms of the manufacturing strategy, so far we've used CDMOs, and we are going to continue to use CDMOs in the near future. There is one thing that we need to be mindful of is that there is significant, overcapacity. So it's not a problem to find CDMO today, but the flip side of that is that some of them are very fragile financially. So we have no intention to internalize manufacturing in the short term, but we have to be very careful about the CDMO we decide to work with, because once you decide to work with your CDMO, most of the time you decide to work with them for a long period of the life of the product, and you cannot change easily in the meantime. And then when it comes to the process itself, as I said, we've made sure that very early on from the get-go, we were working on a process for 06 and 420, which is not exactly the final process because we need to do some scale up. We are going to change a few things here and there, but which was going to be very close to what the final product will be. So we don't expect, in other words, that there will be such a meaningful difference in between the process that we will use for phase 3 and the process that we've used for phase 1-2, especially for 20. For 06, it's a different story. That's a program that we started much earlier and for which there is more, let's say, fixing to do in moving into phase 3 process.
Got it. Okay. Excellent. And just one or two more questions. In terms of the cash runway and the funding needs, what are you funded through? Are you capitalized to finish your phase 1-2 programs in 6 and 20?
Yeah, that's the idea. We are financed until, let's say, sometimes in 2027. But importantly, the mandate that we were given by the Series B investors is to deliver the clinical proof of concept on 06 on 20. So if we are delayed for whatever reason, we will need to find, without raising more money, we need to find ways to prolong the cash runway to make sure that we can deliver on this mandate.
Got it. Okay. And one last question that we like to conclude these sessions with. What do you believe investors may still be missing about SparingVision story?
No, I think you've captured everything. I think we are in a very good place. We have the right skill set on board. We have the right investor syndicate. We have the money to deliver on at least the two most advanced products, which, as I said in the beginning, are clearly first-in-class therapeutics, which have both very, very significant, commercial potential, which I believe is one of the main problems of the, AAV gene therapy space today, is that we've seen some very good clinical success, some very exciting clinical success, but with one exception, being Zolgensma from Novartis, there has been only, let's say, mostly, commercial failures. And we believe that our kind of biology will be able to solve that ROI equation that I was referring to in the introduction.
Got it. Okay, excellent. Well, I think this brings us to the end of our session. Again, thank you, Stephane. Thank you, colleague. We appreciate the time to join us for this installment. Thank you.
Thanks for having us.
Thank you.
Bye-bye, Daniil.
Thank you so much. Thank you.